Peterson Creek has long been a thriving ecosystem and a popular destination for visitors. Located in the Atherton Tablelands, the creek passes through rainforest, farmland, and the Yungaburra Township. But after heavy rainfalls, the water turns murky, and recent signs of declining water quality are becoming harder to ignore.
To understand what's happening in Peterson Creek, we first turn to Yungaburra Landcare Group, a local community organization that has been restoring creek banks and local habitat since 1998.
In recent years, we have wondered about the health of the creek and decided that it needs further investigation. We were focusing on the kinds of water parameters that might affect platypus. We also looked at coliform bacteria and enterococci, another kind of bacteria, and the counts of those were so high that they were practically off the scale, far higher than the national guidelines for safe swimming. The water quality is not good where the creek has run through agricultural land. Upstream, there are several farms with quite a lot of cattle on them.
Thanks to Landcare's extensive research, they have found high levels of fecal bacteria, as well as an overall decline in water quality. So the real question is, where is this pollution coming from? We've come up with a few reasons why.
One is agricultural runoff from nearby farms, particularly cattle waste or excess fertilizer being washed into the creek after rainfall. We also considered the combined effects of land use and rainfall, as runoff carries pollutants from soil livestock areas and roads into the creek and degrades water quality. To investigate these ideas, we used GIS mapping, water macroinvertebrate sampling, and soil analysis to identify potential contamination patterns. For our water sampling, we focused on aquatic macroinvertebrates, which are small organism-like insect larvae and worms that can be seen with the naked eye.
Macroinvertebrates can give us a lot of information about water quality. They have often their developmental stages in water, and that's why they are responding very nicely to changes in water quality.
In order to determine where the contamination might be coming from, we wanted to sample along the entire creek. And in order to do that, we used Geographic Information Systems, or GIS mapping.
It is a super duper tool because we can put maps, various maps on the map of the creek system we are working at, and we can see the land use, we can see slopes that contribute to groundwater flow, and we can extract some information of potential contaminations where they could come from.
We ended up selecting 7 sites along the creek, including Frawley's Pool and Platypus Viewing Platform in Yungaburra. 2 of the sites that we selected were near the source, which have never been sampled before, and taking those samples allowed us to see the water quality at the very start of the creek and then compare it to sites downstream. At each site, we also focused on different parts of the stream, including the edges, the deeper pools, and the faster flowing riffles, to get a more complete picture of the whole ecosystem. Getting to these sites, however, wasn't always easy. Sometimes it meant going through thick vegetation to reach remote areas. That meant lots of slips and slides, wait a whiles, and leeches along the way, as well as getting stuck in the mud.
Once we got to the creek, we put our waders on and got straight to work. Our first order of business was collecting the macroinvertebrates. For this, we employed the stomp and sweep method. This consisted of one person standing upstream and stomping their feet, disrupting sediment on the stream bed. Someone downstream held a net and collected all the sediment displaced by the stomper for 30 seconds. After macros were collected, the sample was poured into a petri dish and sorted through to find the macroinvertebrates. Macroinvertebrates were identified to family using a microscope. Some macroinvertebrates are more pollution tolerant than others. We used a sensitivity index chart to assign specific values of their tolerance level to each family and got an average for each sample site.
To analyze our data, we calculated the Shannon Diversity Index using population counts for all identified families. The SDI, or Shannon Diversity Index, is used to quantify biodiversity by measuring species richness and evenness. It spans from 0 to 3, with 0 indicating the lowest level of biodiversity and 3 indicating the highest level. The highest SDI values were at Frawley's Pool, which is downstream, and South Arm, which is at the source, while the lowest was at Platypus Viewing Platform. Although SDI slightly decreased downstream, there was a considerable fluctuation across sites. Most macroinvertebrates found were low sensitivity, with no clear pattern. This suggests that contamination and declining water quality is widespread along Peterson Creek.
This may be due to local habitat conditions such as vegetation, creating pockets of diversity rather than a clear upstream-downstream trend, and tributaries feeding into the creek that introduce or dilute pollutants.
Along Peterson Creek, we also took soil samples. At each site, we took 4 soil cores and mixed them together. We brought these back to the lab to analyze under the light microscope. We assessed the microbial composition of the soil. This meant counting the bacteria, fungi, and organic soil clusters that were visible under the light microscope. Microbe abundance helps assess soil quality, where extreme bacteria levels and less fungi indicate poorer conditions. Our soil data shows bacteria increasing and fungi decreasing downstream, supporting the contamination trend. These findings are important because declining water quality can reduce macroinvertebrate diversity, the base of the food web, and impact species like the platypus that rely on them for food.
We suspect that almost all the creeks on the tablelands that flow through agricultural land and urban land are going to be fairly poor in terms of water quality. Peterson Creek could be a very good example for other creeks in the tablelands and beyond, because if you can fix Peterson Creek and find a mechanism for doing so, then perhaps that can be applied elsewhere. Possible ways to fix water quality? Well, one of the obvious ones is to increase the amount of riparian vegetation. That is, protect the creek banks from erosion and put in a decent belt of trees and other local species. And perhaps, with goodwill from the landowners and farmers, with some support from various government levels, we should be able to go some way towards improving the quality of the creek water.